Welcome back, everyone. In this explainer, we're picking right up where we left off, diving back into the absolutely incredible world of adult telomerase-positive stem cells, or aTPSCs for short. Now, last time, we laid down the foundational science. Today, we're taking those facts and seeing what they actually do in the real world. We're going to look at some truly awe-inspiring clinical applications where these tiny cellular heroes are literally battling some of the most devastating neurological conditions out there. It's a high-stakes, dramatic journey, and it is all happening at the microscopic level.
Okay, let's dive right into this. Here is our roadmap. We'll start by meeting the cellular heroes, check out the aTPSC superpowers, walk through the multi-pronged deployment protocol, look at the fight against stroke and TBI, the battle with MS and ALS, and finally, we'll wrap up with the final verdict.
Section 1. Meet the cellular heroes, grasping the microscopic scale.
So, as a quick refresher, what exactly are aTPSCs? Honestly, you can think of them as hibernating heroes living deep inside the connective tissue of our own bodies. They are quiescent. That just means they sit there, perfectly quiet and inactive, basically just waiting for a signal that damage has happened. And what makes them so special is they can form cells across all germ lines. That means they can transform into pretty much whatever cell type your body desperately needs to fix itself. And the best part? Unlike embryonic stem cells, clinical studies show these guys do this without ever forming teratomas, which are those dangerous tumors. So you're getting pure, targeted, completely safe potential.
But to really grasp how they do this, we need to talk about their size. Let's use a sports analogy here to make sense of the microns. The absolute smallest ones are the totipotent stem cells, or TSCs. These measure an incredibly tiny 0.1 to 2 microns. Picture these as little, tiny marbles. Moving up a bit, we have the pluripotent stem cells, the PSCs, sitting at 6 to 8 microns. Let's call those our baseballs. Then we've got the lineage-specific ones, like mesodermal stem cells, or MSOSCs. Those are 10 to 12 microns. Think of those as volleyballs.
Now, to put that entirely into perspective, let's look at standard stem cells, the ones you hear about all the time, mesenchymal stem cells, or MSCs. If our aTPSCs range from tiny marbles to volleyballs, MSCs are massive basketballs. They're huge, measuring anywhere from 15 to 30 microns. And as you're going to see in just a minute, the tiny marble-like size of those TSCs is exactly what allows them to pull off biological feats that the giant basketball MSCs literally cannot physically manage.
Section 2. The aTPSC superpowers, the ultimate biological toolkit.
Okay, so these cells basically have 3 distinct superpowers. Number 1, homing. When they get introduced into the body, they actively migrate. They basically home in like a missile, straight to the sites of tissue damage. Number 2, once they actually reach the battlefield, they respond to local cues. The damaged tissues put out these specific exosomes, which essentially act like blueprints telling the aTPSCs exactly what type of cell they need to become to fix the problem. And number 3, which is absolutely crucial, there is zero risk of rejection when using donor TPSCs and PSCs. Why? Because they literally lack the surface markers, the MHC class I and HLA-DR markers that usually trigger the immune system to attack. They're practically invisible to the body's defense mechanisms.
Section 3. The multi-pronged protocol, deploying to the battlefield.
So how do we actually get them into the body? Well, the treatment approach uses different cells for completely different jobs. It's a multi-pronged attack. Step 1 targets the central nervous system by administering those tiny marble-sized TSCs straight through the nose, an intranasal infusion. Step 2 goes after systemic whole-body tissue damage. For this, they take the larger baseball and volleyball-sized cells, the PSCs and MSOCs, and infuse them intravenously right into the bloodstream.
And this brilliantly illustrates exactly why size matters so much. Think about how they completely bypass the notoriously stubborn blood-brain barrier. Because those TSCs are only 0.1 to 2 microns, are tiny marbles. They don't need any intense, high osmolarity drugs to smash their way in. They can literally slide right between the mucosal cells of the olfactory epithelium in your nose, travel straight down the olfactory and optic nerve pathways, and totally bypass the blood-brain barrier to gain direct access to the brain and spinal cord. It's absolute genius.
Section 4. Fighting Stroke and TBI, Dramatic Case Studies.
Now, when we look at the stroke case studies, they reveal something totally fascinating about how our own bodies prioritize healing. In stroke case A, a patient had 4 treatments and saw a moderate solid regain of function in their left arm and leg. But then look at stroke case B. This patient had right-sided stroke symptoms, sure, but they were also suffering from agonizing trigeminal neuralgia, basically incredibly severe facial pain.
After the treatment, that intense pain was completely wiped out for months, but there was barely any reversal of the actual stroke symptoms. The researchers realized that once these cells go in, the body actually hijacks them to treat the most intense pain or life-threatening issue first. So to fix the stroke in case B, they would need vastly higher numbers of stem cells because the body was just so focused on eliminating that pain.
The traumatic brain injury data is just as dramatic. Here we have a patient dealing with quadriplegia, entirely below the base of their skull, along with heavily decreased cognitive function. After just 3 targeted treatments using that intranasal and IV combo protocol, this patient experienced a functional reversal. They were able to slightly lift their forearms, use their fingers to actually type on a keyboard, and even raise their thighs enough to use a stationary bicycle. Now, when you were talking about quadriplegia, that might sound slight on paper, but for the patient, it is an incredibly meaningful return of function.
Section 5. Battling MS and ALS, the compliance conflict.
Here is where human behavior unfortunately collides with the science. In the trials for multiple sclerosis, out of 3 participants, 2 showed absolutely zero response to the treatment. Why? Well, because they completely failed to follow the informed consent rules. During the treatment window, they drank alcohol and smoked tobacco. The clinical data on this is unforgiving. Those substances literally kill freshly isolated, activated telomerase-positive stem cells. If you break the protocol, you are killing off the microscopic heroes before they even step foot on the battlefield.
Now, what's really interesting about this slide is the third MS patient. This is the guy who strictly followed the rules. Before treatment, this person had decreased cognitive function, was fully confined to a motorized wheelchair, and needed a ventilator to breathe 24/7. 3 months after the first treatment, off the ventilator entirely. 3 months after the second treatment, his cognition was fully back, he was out there driving a handicap-accessible vehicle and walking using bilateral crutches. It is an absolutely miraculous clinical recovery, and it was entirely fueled by strict, unwavering adherence to the protocol.
The ALS data, well, it presents a really unique challenge. ALS is just relentlessly aggressive. So the researchers found they had to culture massive numbers of cells outside the body. We're talking between 100 million and 1 billion cells for every single monthly treatment just to fight the disease's progression. But check out this timeline. A patient diagnosed with ALS back in 2013 started intensive monthly treatments in 2015. Through this grueling, continuous application of aTPSCs, that patient achieved a state of stasis, and they are still alive today, over 13 years post-diagnosis. It requires immense cellular numbers for sure, but it absolutely proves that long-term survival is possible here.
Section 6, the final verdict, hope on the microscopic horizon.
So zooming out, when we look at the broader clinical trials, we're talking 97 individuals treating 18 different severe conditions. The data speaks volumes. The cumulative results show a 100% safety record for transplanting these fresh isolates. And this is key, an 86.4% average efficacy for reversing signs and symptoms, as long as patients strictly stick to the informed consent guidelines. It's just a staggering testament to the vast potential of these cells.
So what does this all mean for us? We are standing on the edge of an entirely new frontier in targeted regenerative medicine. If these tiny, microscopic, marble-sized heroes can sneak past the blood-brain barrier, home straight in on tissue damage, and literally coax a paralyzed limb to move again, well, we have to ask, what mysteries could aTPSCs unlock next for incurable neurological diseases? Could the answers to our most devastating ailments literally be sleeping within our own connective tissues, just waiting to be awakened? I'll leave you to ponder that. Thank you so much for joining me on this explainer.